Tracy, Niday, and Gabrielle Johnson. They are science faculty at Southeast Community College in Lincoln, Nebraska. And the title of their presentation is How Attached is Your Dog? An authentic research project to bring bioscience career awareness to high school and college students. Please join me in welcoming our speakers.
Hello, I'm just going to go ahead and share my screen here. So we are in the third and technically final year of our ATE project. And today we'll tell you about all the resources that we've built and also that BABEC in California has helped to extend as well. And we're going to focus on some of the partnerships that have made this project successful, and just kind of give you a little bit of background and how we've been able to build on our biotech program, and also engaging with high school teachers and students through this project. So as you guys all know, biotech is very interdisciplinary, and one of the reasons for this project is really just to increase exposure to this interdisciplinary field and bring it especially to high school teachers who may not have had a lot of experience with biotech. And just a plug for our program and kind of how that has led to this project. Our biotech program officially started in 2021, and we do have 3 stackable credentials, all leading to the workforce, and that will transfer into a variety of STEM majors. And so in the background, there's always work to be done with that. And as many of you probably also experience, we have a limited pool of students that find the biotech program on their own. And so as we've worked with our industry, just trying to come up with a way to, again, how do we get the word out? How do we reach students earlier so that hopefully enrollment will increase? Now, this project, most students are doing this in their freshman, maybe sophomore years. And so we don't yet have any numbers of students that would be showing up in our program. But I think all the partnerships that have been built will definitely, and will hopefully will pay off in the long run. So just a little background about the project itself. So increasing awareness, and then the outcomes in the long run would be that more students do pursue biotechnology as a career. And we chose to do this through an authentic course-based research experience that we were able to develop with our industry partner who specializes in animal genomics. The company is Neogen. So we also have a separate DNA barcoding project that we actually learned through an InnovATEBIO workshop that was done a few years ago on doing more undergraduate research. And so that DNA barcoding project has been incorporated into our majors bio class. And this canine project that we'll mostly focus on today, that has been with our college non-majors biology and getting the instructors for our non-majors biology to be trained and also offer this in all of our non-majors bio labs. And then working with high school teachers, not only to give them a workshop in the summer, but to support them throughout the school year with, we have a full-time project coordinator that will bring all the materials and also assist in the classroom. And that really seemed to be something that teachers, high school teachers, were interested in when we wrote the grant application in the first place, because many of them didn't feel confident in their own skills. And so just being able to come alongside them and support them more than just a workshop in the summer. So that's just kind of some background on the project. And so now Gabby's going to tell you more about the hypothesis.
Thanks, Misty. So as Misty mentioned, this project came to be in collaboration with one of our industry partners, Neogen. It's also based on this Von Holt paper cited on our slide here from 2017, looking at kind of dogs versus wolves and DNA inserts on chromosome 6. There are actually 3 different locations that were presented in this paper, which works out great for us because as Misty mentioned, we're working with different levels of students. So like a Gen Bio student or a high school student, we'll just look at one location. A biotech student or a genetic student, we might look at all 3. So that's been great. So the paper was talking about if dogs, for at least 2 of the locations, if they have extra DNA in these locations, is this extra DNA linked to more social behavior? So defined as hypersocial behavior, it's not quite like friendliness per se, but kind of how attached the dog is to their owner. So this is great because it incorporates, we're looking at DNA evidence, also looking at some behavior studies. And as I'm sure we all know, behavior is really complex. So the larger sample size we can get, the better to look at this. So that's kind of what we're doing with our students is adding to the data set, looking at kind of building off of this initial study. We have linked later, we'll give you a QR code, but we have a video describing our project. And this has been great because we've been implementing this in high schools in our 15 county service area for our college. As Misty mentioned, our non-majors general biology course, biotech classes getting a more advanced version of this. We've also had students doing more thorough research projects. So Tracy had a student who took DNA samples from dogs at a local shelter. And they were testing these dogs thinking like, could this be helpful to know this information for dogs fitting with a certain family? Or maybe could this type of testing be helpful for, is a dog like fit to be a service animal, something like that. So a lot of potential impacts and really exciting projects that we've been able to do with students. And then as we kind of always mention when we talk about this, like, yes, the research aspect is really exciting. Like we do love, really it's become a citizen science project. But I think something like Misty, Tracy, and I are really passionate about is just like giving students and teachers in our area these like really buildable skills, right? So pipetting, sample collection, DNA extraction, PCR, running gels, actually analyzing real data that they're generating. These are really transferable skills. We hope that our students will go into biotech, but maybe they still want to be a biology major, something like that, that's fine too, but they're getting really great hands-on skills that they can apply for a variety of things, whether it's, their continued academic or potential careers later. Then I think Tracy is going to talk about some of our industry experiences.
Yeah, hi everyone. So one of the things that we really like about this project is that it's industry linked. So our partner Neogen, which is an animal genomics company, I like to say they're the 23andMe for animals like cattle, pigs, different livestock, and also they do dogs. So we actually did the workshops for this project at Neogen so that the teachers can be exposed to the industry. And one of the biggest comments that teachers would say is we didn't even know this was in our own backyard. We didn't know this was a career possibility. We've had a lot of teachers say that they have students that they know are going to graduate and not go to college, but don't see themselves getting this type of job, but it could be a potential job for them because they're learning these skills in their high school classroom. So I think working with Neogen has been great, but we also have all these great partners. So EPSCoR, our Nebraska has an EPSCoR center that we work with, and they are actually going to take this project on and distribute these kits that we have made for free to all of Nebraska. So one thing we have found is we have a 16 county area, but as we have advertised for these workshops, word is getting around, and very, very rural areas in Nebraska that are pretty far out are asking to get trained on this technology. And we actually don't have the means to do it with our grant. But EPSCoR, which is Nebraska-wide, will be able to reach this rural population. So we're super excited about that. Mini One is the equipment that we're using, which we really like for high school labs. Don't Panic Labs is a local company that's working on our database. And then obviously Baybeck has done an amazing job of actually taking this curriculum that we developed and kind of making it more streamlined, because they've been doing this for like 30 years. They have great high school curriculum writers. So they have made this into a consumable kit that you can actually purchase. And I think, do we have that later on the slides, Misty? Yeah, so I can show you that link later, but they've done an amazing job. And so working with them has been awesome. So I think one of the big things about this project is like all the partnerships that we've built to really help us out. And then just to expand on that, I would say Misty thinks I'm being overzealous about this, but I really think that getting this ATE grant has allowed us to basically get the momentum and the resources to expand into a lot of other areas for our program. So it's funny because you think you're getting an ATE grant to do one project, but then you go to conferences and you meet with all the InnovATEBIO people and you collaborate, and literally all of these opportunities just started popping up. And it's actually a little bit crazy. So I will go on the books and say that, I mean, it started off with an InnovATEBIO undergraduate research grant, which actually helped us develop this dog project. Then we applied for the ATE grant. And then we just started going to conferences, talking to other people. And so like now, if you look on the right-hand side, Lycor Bio is another industry that in our local area that does fluorescent protein imaging or different fluorescent imaging. And so we've created another industry link cure that is based, I call it biomanufacturing the rainbow. So they bought me a whole bunch of stuff to basically do biomanufacturing. They helped me buy some equipment. So we're starting to create more of these industry link cures. And then we were actually able to work with UNL and apply for an NSF Global Center grant, where we're doing this same model. We're using our resources with all of our teachers, but we're going to move on and do fermentation as it relates to the future of food. So I would say, I know I might be being too zealous about this, but I think that the ATE grant is really, really important for sort of bringing momentum to your program.
Tracy, do you want to mention anything about our new spaces? Our new
Oh yeah, sorry, this is really good. If you look at the bottom of the screen, one other cool thing that happened was our administration saw what we were doing in the community. And because of that, I think that in our industry partnerships, we built this new Sandhills Global Technology Building, which has brand new biotech lab spaces. And after visiting Jim DeClose's lab in San Francisco, which blew my mind, I actually designed the lab to be very industry focused, and my administration pretty much didn't say no to anything. So they said, how many biosafety cabinets do you want in this mammalian culture room? I said 4. They said, okay. They said, how many autoclaves do you want? I said 2, pass through. They said, okay. So we now have these brand new spaces. And I think this will only, help us in the future to grow as well.
Yeah, these spaces are literally Tracy and Gabby moved over there over the holiday break. So yeah, super brand new. Yeah.
Cool. And
well, is this where we get to ask questions? Dion, you can ask for questions, or?
I think we have just a couple more things.
Good.
Yeah,
Fantastic.
I'll just quick go through this. Just to give you some numbers, we have helped out 36 plus teachers. We actually just got the University of Nebraska Lincoln, thanks to Gabby, their genetics classes are going to start running this, which is really cool. So probably the number is probably higher. I bet it's like 3,000 or higher students reached. in our area. We haven't tracked it nationally. Just a quick example of what the data looks like if you run a class of several different dog samples and you do and you look and see, does the dog have an insert on one chromosome? Does it have an insert or extra DNA on 2 chromosomes? We call that homozygous insert, or maybe they have an insert on just one, and that would be the heterozygous kind of shown. It's so weird because I'm pointing it to it with my finger. It's the homozygous insert is that top band where it says 6.7. So, or you can have heterozygotes where it has, you know, one of the amplified products has no insert and one of them does, or it can be homozygous wild type where the dog doesn't have any extra DNA. So we run that gel, each student analyzes their dog, and then that student is also given a survey of 5 questions, and we score that survey. The higher the score of the behavior survey, all the way up to 20, the more hypersocial or attached the dog is. So we then go and we average all those genotypes, the hypersociability score, and we see in the class data, is there a correlation? Yes or no? Is the hypothesis true? And the cool thing about this is, honestly, every class can be different because it's such a small sample size. And then the next thing, we do have this database, which is still a little bit of a work in progress, by the way, which...
So there's upgrades and a new iteration that's coming. It's about half done and should be done in the next month.
Yeah, so we had to do some modifications, but this is looking at everyone that submits their samples. It's looking at, is there a trend in a large sample size? And you can actually look at this, and it's actually refuting the hypothesis. So there, at the 6.6 gene loci, the dogs that have insert, that genotype actually has a lower hypersociability score. So currently, with our current sample size, this says 59, but I think now we're up to like 200 that have actually submitted their data since we had to redo the database, but it's still trending down. So, but this allows the students in real time to go look at the database and say, with this larger sample size, what does the data mean? And it's not always supporting the hypothesis. And then here is the BABEC page, which I mentioned they have, basically made a curriculum that's very high school friendly, user friendly, and it looks beautiful. And they also are selling reagent kits. So we do have a list of all the reagents if you can make things from scratch, like if you want to order your own primers and do all that. But if you don't have time, especially high school teachers don't have time to do that, you can click on their link, like the reagent orders on the left side of their page, and you can request a kit from them, which is great.
And Ying-Su, did you want to offer anything else about the resources that BABEC has?
Yeah, I'll just thanks for that. And by the way, we love working with you guys. This is so fun. I'm just going to show you a little bit for the audience out there what we mean by that. And it's really useful too for not just high school, but if you are teaching an introductory biology class in community college, is super useful too. So we have our resources. If you click on that cute little doggy picture, you get to this page. If you register, so I'm already registered, then you can get access to our lesson and lab resources. And these are all downloadable and editable. So if you don't, we do this as an entry for teachers that don't have a lot of time. They can just take our stuff and use it. It even has lesson plans, for example, minute by minute. And my staff, my staff, right? It's Michael, Elizabeth, who are the curriculum writers and practitioners of our labs. It's tested, it's piloted. And so these are really useful if you want to take a look at that. And we also have slides to go with that. All resources that are free. There's no charge for this. So take it and run with it. We also provide the kits, as what Tracy said. So that's all, for your use and for your however way you want to use them. I must say that we had a workshop recently in October, which was really fun. So Nebraska has come out to California now. And we had a workshop, K9 PCR, for 20 of our teachers, roughly. I don't remember how many came. And it was fun. They really enjoyed it. And they also gave some feedback for your website, which was great. But they all were really jazzed about it. And it is such a nice, open, we get a lot of feedback sort of like, well, it's cookie cutter or it's a recipe. But just science is like that, right? If we have a protocol that works, why change it? But it's the question that we're asking that's changing. So this is really ripe for like, oh, what are all German shepherds like? What are all, labs like? Or what are the guide dogs like? So, it's this really nice project that you can ask a lot of different questions for and still, use the same sort of protocol to answer those questions. But anyway, thank you so much, Nebraska.
Thank you, Yingsi. You guys, what you do is amazing. When I saw that, I was like, the curriculum, I was like, this is the best.
Yeah, so basically we were able to we provided an initial framework and kind of let teachers do with it what they wanted. And then Baybeck has provided a more structured approach there for sure. I see that in the chat, people were asking about
Yeah, so I want to
pictures of the dogs.
What I'll do is I can start at the beginning of the chat, Misty, if that's okay. And if you made a comment, feel free to elaborate. Yingsu, you made quite a few comments about the just different things, but the first one you mentioned was the Association for Biology Lab Education. Did you want to expand on that?
Yeah, I'm just curious because I don't even know what that is. What is
Oh
that?
yeah, sorry. So actually Gabby's the one who told me about this because she was at a NSF IU's conference and there's a lot of people that are involved with this ABLE. And I guess they've been doing this for like 30 years, but it's a conference that's just about teaching labs. So it's going to be in Canada this year. Last year it was at the University of Maryland. But we did a major workshop there, which is where they give you a 3-hour time slot and you teach a lab. We actually did it twice. And then you also get to attend other lab sessions. So everyone is teaching a new lab session and they're all new labs that have recently been created. And then you can publish your lab at the end in a journal. They have a journal article. So that was a really cool experience, not only just to present there, but we actually attended those workshops and got information for our labs. And we were the only community college there, right, Gabby? I can't remember.
A lot of four-year schools really
Yeah.
from around the country, and then various, science representatives from different companies. So we've reached kind of a different audience than we typically do, which was nice. It was a lot of fun.
Yeah, and Lene, you mentioned about ATE networking, a presentation with that.
Well, I just like the fact that you understood how to leverage your ATE grant, and that's what everybody should do, because I relate to that because that's exactly what I did too. And you should never think of it as only one stop and that's it. You should always think about the next grants and who else will be interested. So kudos for that.
We've been listening, Linnea.
Yeah. And also thank you to InnovATEBIO because honestly, we wouldn't have probably gotten our ATE grant unless we went to that undergraduate workshop, that Jim held, and then you guys supplied us with our reagents for the pilot of this. So.
And I will say our industry partner also came to that workshop. And then I think that's when they're like, oh, well, maybe we do have this other project that's interesting that would be interesting to students as well.
May I ask a
Laura,
question?
Laura, you have actually
Yeah.
two questions in the
Right,
chat.
well, I'm gonna call it one question, but I did put questions in the chat. Can you talk a little about I think it's gonna be a safety concern. I know people, it's their pet, but there's probably a safety concern around that. And then also just something about the ethics, if you find out something about your dog through this that was an unexpected finding.
Yeah, that's a good question, Lori. Honestly, we weren't as concerned about the safety, and then Ying Su brought that up when we started working with her. So we do tell, I think Ying Su does, you know, with Baybeck, they have like a consent form that they have people sign to swab the dog. We tell students, really make sure that only the owner is swabbing the dog. So a lot of time you can't just go run to the neighbor's dog and swab it yourself. Obviously you need to get the survey information. So I guess for us, the way that we think about the safety is, you know, we have a video showing how to swab the dog, and it's not invasive. It's just a roll of a brush inside of their jowl real quick. And then we tell them to have the owners swab the dog. That's how we address the safety. I would say ethics wise, like I think the way that we've approached this after talking to Monique Udell, who is the behaviorist at Oregon State, a lot of the news media, when they picked up the story, they say, how friendly is your dog? Which I think is a little bit, as Gabby mentioned, it's not quite the friendliness. So we don't want, when we approach the teachers and we tell them to approach their students, especially the middle school, we have done this in middle schools as well. They don't want to say, oh, I found out my dog's not friendly, right? You can't have a middle school finding that out. So Monique's point of view is like, when you present this to students, it's more of like a spectrum of like introvert or extrovert or independent and dependent. So a dog that is, it's not that the dog is not friendly, it's that the dog is more independent and will do things on their own without the owner. And so approaching the behavior and sort of a more of a objective way is sort of how we frame it. Does that help or answer your questions?
Yes, thank you. And also this is just the coolest thing. And actually, we have a faculty member who's not biotech at MC, but who is usually seen with a service dog in training. She socializes them, and I can just see her picking up and running with this, but she's on sabbatical. I'll let her know when she comes back.
Okay, that'd be great.
And thank you, Laurie. Did you get both of your questions? You said that you added another one. Okay. Linnea, you actually had another question about working with psychology or presenting to the social sciences.
Well, you know, it's interesting. While just sitting here listening to you, I Google scholared this, and it's really, and you probably already know this, is that this looking at the genetics of the hyper sociability, it's related to also studies in humans. There's a lot of work done on this. So I could see where this would be interesting to do with students in psychology or sociology as a way to make biotech even more interesting as a cross-disciplinary type. Hey, you know, submit another grant that you can do an iUse or something. I'm just saying that I didn't realize how much research is done in this area. And then it's associated with even a neurodiversity disorder.
Yeah, it's like William, I can never say that, Misty.
Yeah,
Williams-Barron.
Yeah, so the 60 Minutes did a thing, and I think they deleted the videos, but they actually showed a person with that disorder, and he's like super friendly, like he literally just loves everyone. And so it was really cool when they interviewed him.
I can literally send everyone the paper on that, but it comes up very easily in Google Scholar. So I think this is really can lead to a lot of other things too.
Just put the title in the chat.
Okay.
And I can mention one thing, Linnea. We haven't, we worked with one group, or we had a meeting with a group at UNL. They have like a brain behavior research group. But a couple of semesters, SEC has an animal behavior class that's a cross-listed biology psychology class. And so it's actually just a lecture class, but we were like, let's bring these students in. So Tracy and I co-taught this animal behavior class with a lot of psychology students, so they got some hands-on lab experience. So we've done that a couple of times.
Yeah, and on our SEC faculty, one of the social science faculty is super into dogs, so she's the one who co-teaches that class. So it was a really interesting class.
Thank you. Russ, you have a question?
Yeah, I thought this was really a great presentation, Misty and Tracy, and thank you very much. That's great, and Gabrielle. My question is, I think Elizabeth and I are probably thinking the same thing. You said that Neogen's sort of like the 23andMe, but what's in it for them? And has the relationship changed at all since you've been doing this with them? Thank you.
Yeah, Neogen is probably our biggest supporter of all the companies. And as you know, Russ, with working with companies, a lot of companies want the short-term payoff. And so they want things right away. Neogen has been taking our practicum students since the very beginning. And so a lot of our practicum students have ended up working there. I think the relationship with them continues to grow, and they're playing the long game here. So the CEO or the head manager, Niraj, basically realizes we need to get into the younger groups, and he's really amazing because he's doing it not only for his company, but he realizes for biotech in our region, this needs to happen. So I think he's also, Misty, isn't he the chair of our trade organization now, right?
He's the chair of our workforce leadership team, and he also is on the board of our trade organization.
Nebraska. Yeah, so I think we're lucky in that a lot of companies don't maybe have the time or resources, or not willing to commit the resources to play the long game. But I think we all know here on this call that the long game is actually super important to support the short game as well. I don't know if Misty, you want to add anything?
Yeah, on the Neogen side, in terms of tests that they offer, they do already include this particular gene, these 3 gene loci, in their vet diagnostic types of panels that they offer. At least that's the last that I heard. But I think they added that, but they are somewhat interested in when there are a larger number of samples, does the hypothesis hold true? I don't think it's like super important to them, but like Tracy was saying, they're more interested in more students learning about biotech and the things that it's capable of doing.
And Elizabeth, you had a question along the same lines.
Yes, I was just interested in how Neo fits into it, but that totally makes sense, and I'm just thrilled that a company is interested in the long game as well as the short, of course.
Yep.
Thank you.
Maggie, you have your hand up.
Hi, thanks. This is great. I'd heard about this from Ying Su, and it's really nice to hear more details, and it really gets me excited to introduce this into our high school student summer workshop. So my question is, once we run the experiment, are you collecting the data from high school teachers, from faculty, as they go ahead and incorporate this experiment for your data set on your website? Should we be sending that to you afterwards?
Yeah, so the database is currently available for students to add data to. So I'll have to drop that link. I can drop that link directly in the chat. And on let me switch back to our presentation. This let me just show you a couple things on the current website. has data that's updated in real time, but it also has, the students will upload pictures of their dogs. So all the dogs are here. My internet's a little slow, but so all the dog pictures are there and available. In the next iteration of the website, then the goal is that when you hover over a picture of the dog, then it'll pop up with that dog's result. So, and it is nice also in terms of like, we don't have any like student data directly, it's just the dogs. So that does make it a little bit easier to navigate when we're working with high school students, to not have any data attached directly to the student. But back over, let me switch back over here.
Yeah, so it's a Google form that the student directly fills out with their data, because we thought it would be too much work for high school teachers to fill in, to put in the data themselves, and or us fill in everyone else's data. So that's a little bit of a risk, is that the student is inputting their own data.
Yeah, and so that's why also in the next iteration of the database, there'll be a few more checks and balances to make sure that all the data that comes through is the correct data to flow into the database. But I can drop a link to that Google form. It is, if you use this QR code and go to all of our resources that are Google Drive folders, that link is in there, but I'll get it here and I'll drop it in the chat.
Great, thank you.
Okay. Thomas has his hand up.
Yeah, so I have two questions. One is going back along the ethics line to IACUC approval, because I know I'm at LaGuardia Community College, City University of New York, CUNY, and we have we're one of the few in the area that has a vet tech program for years. And I'm a reproductive cell biologist. And so when the students there even did spay and neuter for teaching purposes, I was like, oh, you're throwing that out. I would love to do reproductive research with the students. And there was the vet tech director, Tom, was like, that's research. You have to go through IACUC approval. So even if you're swabbing the animals and so on, how do you get approval for that from your institution? That was my one question. The other one is, with these regions that you're amplifying, do you then go and sequence it? Because there might be sequence differences between each of them. And then it'll be interesting to see whether they have those inserts, but then what's the sequence? And is there any mutations in the sequencing that lead to maybe more friendliness and unfriendliness? And then coming from New York City, and then looking at, if you do it with dogs in a rural environment, has urbanization even made them more friendly or so on? So it'd be interesting to compare rural and urban environments. And I coordinate our non-majors, I call it baby bio, and I'm looking at ways to make more interesting, fun labs. I think this is absolutely wonderful. When I saw canines, that's why I was like, I registered right away for this, because it's not a research project you see very streamlined. So I'm very interested in this. Plus my sister's a vet tech now, and she was vet tech for a while. So I find this very interesting, and it's very, very, I think, applicable to a lot of people, right? Because a lot of people are dog friendly and owners. But I just want to know, like, how do you deal with the IACUC approval? And if you're dealing with multiple institutions and citizen science, how do you have to have any protocols in place? Because they are then, going to be subjects, even if you swab them.
I will say that when our project was being reviewed for if it even needed sort of like that animal type of research oversight, that didn't come up as an issue since it's non-invasive. So that's all I have to offer. We don't have our own review board. It was done through UNMC, and they didn't have an issue with it.
And what about
we haven't done any sequencing. I do think that the sequences are different, by the way, because I've played around with the PCR conditions. Because we do get some interesting things where if you run, especially, it's a Kelex bead extraction. And sometimes we have our students do pure genomic DNA extractions because Helix, it's not super pure, but even with pure genomic DNA, when you do the PCR in different dogs, slightly adjusting the annealing conditions can increase the amount of insert versus wild type or vice versa. It changes the intensity. So I haven't looked at the sequencing, and we have a nanopore now, so maybe that would allow us to sequence it pretty quickly. But my hypothesis is that there are single nucleotide mutations where the primers bind, because different dogs, the intensities are different at different annealing temperatures. Some dogs, you'll get the same results each time. And some dogs, if you change the annealing temperature, it changes, like there's a whole bunch of insert and a little band of wild type. And then, by decreasing it 1 degree or 2 degrees, it will change that dramatically. So I do think there are mutations. We just haven't looked at that.
And then finally, Ying-Su, you have you put a lot of comments in the chat, and then also a few links. Do you want to elaborate on some of those links that you shared?
Gosh, where do I start? I think I put the link for the 60 Minutes. I believe it's CBS News. That's the coevolution one. They've done a lot more research lately, recently, that beyond that original paper. And if you look at the original paper, the sample size aren't large.
Yeah.
And there's really three that they focus on, although you can see a whole list of loci that they look at. And they also looked at 3 different types of behavior that they initial ABS or HYP and things like that. And they have different correlations to each locus. So that's also another piece of research that you can always dig into.
And it's pretty like another just level of complexity is that we did, in talking to Monique Udell, because I'm not a dog behaviorist, we're using the dog survey because it's easy to implement. It's 5 questions from a national dog survey called the CBARC. However, in that paper, they also have 2 different behavior tests, a dog puzzle and a proximity test. And Monique says that she doesn't fully trust those survey questions because she hasn't had time to vet them. She trusts them in that they have been part of this survey for a long time, but they haven't specifically been vetted for hypersociability in a statistical test. So she's working on that, and she's actually interested to see our data for that reason, because we're kind of doing that work for her. But there is also, we have the directions for the behavior and the food puzzle on our resources. And I know, like, I think it's in Missouri, Elizabeth, there's some instructors that are sorry, what?
Monica?
Oh yeah, sorry, Monica. I don't know why I'm saying Elizabeth, probably because Elizabeth did the curriculum. Monica, she has her students do the behavior test, and we do have a place for that on our database. We just find with high school students, they don't have the time to do that. But that's another level of complexity you can add if you have time.
Well, and one aspect of that behavior test is that it is asking for someone who is unfamiliar to the dog. And that is something where some people aren't comfortable with that. Like, someone outside of maybe the people the dog is familiar with, that does bring up some safety concerns there. So that, I think, is a little bit of a limitation for some of the high school teachers in implementing the behavior test. The puzzle, we haven't tried that one. Monica has, so and maybe that one would be a better option.
So I'll just add in, I think you should make Dave Mikolos, who's running the genomics hub, have a meeting so that all the ATEs in genomics could explore further additional grant opportunities and research opportunities. So feel free to tell him that I told you to jump on his case.
Okay.
Yes, he had a lot of questions for us at the poster session at the ATE meeting.
You know, he's pushing hubs, so he could have the first hub meeting.
And Monica, you had a couple of comments along with a question. Did you have your question answered, or did you want to elaborate? Is she still on? Oh yeah.
I am. I'm sorry. I'm sort of in a place I can't talk really well. I'm fine. My questions were answered, and I'm happy to talk with anybody about the puzzle testing. We started implementing that last semester in addition to the face-to-face behavioral testing.
Yeah, so Monica, maybe you want to speak a little bit more. You kind of turned this into like a whole semester, or at least part of a semester project, just my understanding.
Yeah, it's a whole semester cure now. We do some work with, scientific literature and looking at domestication and just sort of the evolution of dogs and wolves. And then we visit a wolf sanctuary so that they can sort of see these wolves up close and personal and really kind of get a good vibe on how different wolves really are. And then we start the testing. Students do their own dogs first. They build the puzzle boxes in the lab, and we go through all of that, and they test their own dogs first. And then we hold a dog clinic on campus for our faculty and staff to sign up to bring their dogs for testing. And I do make a point that if they feel that they have a dog that's ever been aggressive, they cannot participate. And we
Do you have
require
a
them.
syllabus for your course, just out of curiosity? And
Sure do.
I think I posted it also on our resources page as well, Monica.
Okay, great. Thank you. Thank you very much.
So much.
So that's what we do. So we do dogs from our campus community, and so my campus community is very, very excited about it. And we actually have a wait list of people wanting to get in and get their dogs on campus for us to do some testing. So it's been really awesome. And I'm going to start to incorporate also some coat quality insertion genes too, for us to start to look at to expand it a little bit more.
Cool. Yeah, she's taking it and run with it. That's awesome.
Yeah.
I was just so excited when you guys came up with this, because I was really looking for something, animal related and something my students could really embrace. And I can't thank you guys enough for getting this out there, because it's really been just so phenomenal. And my students are I have students signing up for the class just because they want to do the dog research, and it's majors level bio. So they have prerequisites they have to meet to get there. So again, I thank you guys for just how much, wonderful things you guys have been doing with this.
Terry, you have a question that you placed in the chat.
I was just curious if it's possible for us to send samples of our own dogs to have them analyzed. Both of my dogs are rescues, and I'm guessing different things about them based on their behaviors, but I don't know their history. And one of them is clearly has trouble with certain people, certain specific people, but loves everybody else. And so I assume she was mistreated by someone that looks like those people. And the other one loves everybody. So I think she was only neglected, not abused. But it'd be fun to know.
I do have some extra work studies. I guess if you're interested, we could send you some swabs and then run your samples.
Looks like Yingsu's offering as well.
Oh, Yingsu.
Great. Yeah. No, I would love to do that. I've used Embark, which is from Illumina.
That's what I've
done the age test on both of them, and done the breed test. And so I have a good sense of who they are that way, but it's fun to know more about them.
So everybody, that the amount of money and research into dog health, cat health is huge. So how many of you out there I will put in chat because, this is a great way on how to get to people to learn about biotech. So another example is I now have 2 dogs on the monoclonal antibody injection for treating pain associated with osteoarthritis in dogs. How many of you knew about this?
Is that made? I think that might be made here, Linnea.
Yeah.
What is it? Zoetis?
No, well, this is Libra. So they
but-
had originally done the studies in humans, the idea of this monoclonal antibody, but it had a side reaction of acute psoriasis. So now the company is just doing dog and cat products. And I will tell you, this has totally made my 12-year-old dog like a new dog. But, this is a great way to get people to understand the value of biotech is through their pets. Okay, that's may sound cheeky, but whatever it takes.
Yeah. I will say when we had our grand opening, we somehow brought up the dog project, like one of our grant assistants. And then I found out later from our VP that was the people we were talking to was like a senator and his wife, and then the president's wife. He's like, you did really good so they're going to bring in their dog swab. So it does seem to like open up conversations with people in a different way, I think.
Yeah, so I put in chat the Librela is the monoclonal antibody that I now have my dogs injected with. Now they don't need Rimadyl anymore.
Yeah, it's made by Zoetis. I'm pretty sure that's made here, Linnea, because
Yep, it is.
Zoetis just opened up. So a monoclonal antibody manufacturer in Lincoln. And the interesting thing about that is it's FDA regulated, not USDA. So our companies are having issues because the people are used to working in a USDA culture, and shifting their mindset to FDA has been difficult.
Well, that this is what it was started for. They were starting on humans, and they may eventually go back to humans. Do you know what that would do if we could do this for humans? Eventually, it's just a lot of side effects, but I think this is a great way. I'm using this to educate students about monoclonal antibodies. But I will tell you, it is a miracle drug for my 2 dogs who have arthritis.
Well, I'm going to take this time to thank all 3 of our presenters, Misty, Tracy, and Gabrielle. That was such an informative and engaging presentation. Whenever we talk about our pets, that will definitely be interest, just like our kids. If you would like more information on this ATE project or our up and coming events, our next project talk will be February 7th. And I will put the link to the project, the upcoming events in the chat. Cassandra already put the link for the evaluation. Please complete the link, and in the event that you have to jump off really quick, that will get emailed to you as well. This presentation will be on our InnovATEBIO website. Again, thank you to everyone who spent your little bit of Friday with us. We wish you well and a happy and safe weekend. Thank you.